Bismillahirrahmanirrahim. Hello, all the participants, members, and organizers, distinguished guests of this International Conference on Biosciences 2021. I am highly thankful to the organizers of this international conference to provide me the opportunity to speak out here in this distinguished platform. My name is Siyab Alam, the student of BS Biotechnology in the University of Malakand. I have prepared this poster presentation with the guidance of my respected supervisor, Dr. Tariq Khan, which is the evolving strains of Carbapenem-Resistant Enterobacterales. In this very presentation, I would like to proceed sequentially. First to talk about the introductory words about the antimicrobial resistance, and then the mechanisms, etc., and then the alternative option which might help us to tackle the menace of this evolving strains of Carbapenem resistant Enterobacterales. So before proceeding to start all about this, the present details of this presentation, I would first like to introduce and define what really resistance in the pathogens is, and what is the buzzword currently in the world of antimicrobial resistance and superbugs. So first of all, I would like to define what a superbug is. The nightmare of evolving superbugs. Superbug is another name given to the bacteria or a fungi that have developed the ability to resist commonly prescribed medications. Antimicrobial resistance occurs when microbes evolve certain mechanisms that protect them from the effects of antimicrobials. The term antibiotic resistance is a subset of antimicrobial resistance (AMR), which applies to the bacteria that becomes resistant to the antibiotics. While any germ may become a superbug, bacterial and fungal strains that routinely infect humans, animals, and crops are most likely to do so. According to the Center for Disease Control and Prevention, the CDC, about 2 million people acquire vulnerable infections because of these resistant pathogens, and about 23,000 people die annually because of the resistant strains related infections. Now, what is Carbapenem? Carbapenem is considered a last resort antibiotic against the resistant strains, which is a class of highly effective antibiotic normally used for the treatment of high-risk bacterial infections. They are usually reserved for known or suspected multi-drug resistant bacterial infections. Carbapenems are those class of lactans class of antibiotics which kill bacteria by binding to the penicillin binding protein, and thus inhibiting the cell wall synthesis of bacteria, coming the bacteria to an end. Compared to other antibiotics like penicillin and cephalosporin, these are also beta-lactam antibiotics. So these agents individually exhibit a broader spectrum of activity. But recently, with the emergence of Carbapenem-resistant Enterobacterales, a new class of antibiotic which mainly consists of Klebsiella pneumonia and Escherichia coli, they have posed a new threat to the world by imposing certain mechanisms and increasing the morbidity. General information about the Carbapenem-resistant Enterobacterales. So CRE or Carbapenem-resistant Enterobacterales are a major global health threat. They have been ranked by the World Health Organization's priority list as among the top 3 multi-drug resistant pathogens which needs solutions. Infections caused by CRE are challenging to treat due to limited number of antimicrobial options. This leads to significant morbidity and mortality worldwide. This poster depicts the use of silver nanoparticles as an alternative to tackle or to cope with this global menace of antibiotic resistant strains. Now, moving down, I would like to tell about the general information about the CRE, that how they developed the mechanisms and how they evolved the mechanisms to inactivate or to cope with the application of antibiotic. There are three main mechanisms. For example, the first one among them is the Purine mutations. So what are the Purine mutations? Purine mutation means that there is there is certain shift in the purine expression. Purines are the proteins and the surface which regulates the in and out of the certain ingredients inside the cells of the bacteria. The number second is the efflux pump. So efflux pump is the mechanism which the bacteria switch on and off occasionally to deal with the osmolarity situation. So if the bacteria takes the antibiotic in a high quantity, then the bacteria evolves certain mechanisms to efflux out the antibiotic which it has taken already. Thus the reduced concentration of the antibiotic renders it to be unable to kill the bacteria. The third mechanism by which the bacteria adapts itself with the antibiotic or render it incapable of killing it is the enzyme production. So bacteria produces a number of enzymes. These bacteria like extended spectrum beta lactamases production and certain other enzymes which enables like certain plasmid mediated or chromosomal enzymes which has certain hydrolytic activity which hydrolyzes the antibiotic as it inside the bacterial cell. Now coming towards the major infections caused by the Carbapenem-Resistant Enterobacterales. So these bacterial infections by Klebsiella pneumonia, specifically by Klebsiella pneumonia and Escherichia coli, are recently and they are most commonly observed in people who have been for long time in the hospital. So hospital associated infections like then the pneumonia, meningitis infection, then the liver and spleen problems, and certain type of skin infections. Symptoms of different CRE infections are shortness of breath in case of pneumonia, then pain with urination when there is the urinary tract infection. In case of the skin infection, there will be the swelling of skin and pain sensation. Belly pain in case of the liver and spleen infection caused by these resistant strains, and stiff neck and reduced consciousness in those patients which have been recognized with the meningitis infection. How resistant to these bacteria spread in the community? So this is a schematic presentation which can tell us about the spread of these infections. So in the very first, the mixed bacteria population which infects a person, then we administer certain antibiotics to kill them. Antibiotics kill only non-resistant bacteria, while the resistant bacteria by applying certain mechanisms it rules out the antibiotic and render it unable to kill it. Then the resistant bacteria multiply and grow inside the patients, and then it spread to the nearby people, and thus enabling its spread in the community. This is an statistical analysis which is showing the annual increase in Carbapenem-resistant enterobacterales occurrence across the globe. So this is an alarming situation like we see in the very first graph that from the 2011 to the 2017, on the x-axis of this graph, and the percentage of the resistance. So it is clearly indicating that as a whole there has been an increase in the probability of the infections worldwide. In the very second graph, we can see that the estimated cases of the estimated cases of CRE in hospitalized patients. So from 2012 to 2017, there has been a significant increase in the patients who have been for a long time in the hospitals. So owing to such a problem, a global health threat, the WHO has also called for opting for finding different solutions to these infections. So in this presentation, I have devised this plan and showcase the silver nanoparticles as an alternative remedy to deal with these infections. So this is a schematic presentation in which the silver nanoparticles has been shown. So silver nanoparticles, certain recent studies conducted have proposed the use of biologically synthesized silver nanoparticle as an alternative strategy against CREs. In this strategy, silver nanoparticles, when used alone, showed antibacterial activity against Carbapenem-resistant strains. Then silver nanoparticles used along Carbapenem showed synergistic activity and promising results to tackle these resistant pathogens. Like in this very schematic presentation, we can see that when the Carbapenem alone is applied and the bacterial cell takes it, so the bacteria activates one of its mechanism, that is Efflux Pump, and expels the antibiotic out of its cell, and thus survives the application of antibiotic. But when these antibiotics are combined along with the AGNPs that are synthesized by either way, maybe by the chemical way or by certain biosynthetic pathway, so these nanoparticles along with the Carbapenem antibiotics, when they are applied inside the cell, then these these bacteria are then unable to rule out or to expel out these antibiotic, and owing to the antibacterial activity of the AGNPs, the antibacterial activity has been seen, and these bacteria which was previously resistant to the antibiotic, now it shows a wider range of activity against these bacteria. And thus I propose in this very presentation that silver nanoparticles can be a potent and an efficient alternative way to tackle with the multi-drug resistance, and specifically Enterobacterales that have resistance to the Carbapenem, the last resort antibiotic. So by applying these strategies, the world can overcome by the synergistic activity, the world can overcome the resistant strains, and we can tackle these bacterial strains. Thank you.